obviously there are major implications about finding virus in multiple regions of brain and specific cell types. So we're quite cautious about those observations and have taken a number of assays to support or confirm that, cell type specificity. As far as replication in the brain, Nihal, we have right now indirect and are pursuing direct evidence of replication competent virus in the brain. The indirect evidence is that the sequencing results that I showed you show evidence of minor variants in the brain, and sometimes not minor variants. Actually, they're nearing equal distribution of variants with non-synonymous mutations that we detected in the brain that we did not detect in the respiratory tissue, suggesting that ongoing viral replication does take place in the brain and that there actually is some evolution of the virus in the brain. We have tried to grow virus from the brain. We're not done. We're going to persist. So far, we have not definitively accomplished that, but it is something that we continue to pursue. Tissue preservation and short postmortem intervals to be able to glean the observations that we've had. But the observation in an N of 2 is that there were viral variants in the brain, populations of viral variants in the brain that were not detected in the respiratory tract, suggesting that there's seeding of a virus in the brain, there's ongoing replication in the brain, and that we obviously know there's an RNA virus that's error prone, and that as it replicates, there are variant populations that are produced, and through the very sensitive sequencing technique out of the Boritz lab, we're able to detect variants in the brain that were not detected in the respiratory tract, suggesting that there is replication there, and perhaps that there's a particular immune environment there, and this is speculative. Obviously, that's an immune protected site that would allow for viral replication and evolution of the virus. You may recall that our final study aim was to determine if SARS-CoV-2 evolves in different anatomic compartments within the same patient. To address this question, the Boritz lab performed high-throughput single genome sequencing to analyze spike gene variant sequences from a total of 46 tissues from 6 individuals. Sequences from P18 or patient 18 are shown on the left, which were identical across all anatomic compartments tested. P19 on the right, in this patient we detected a single synonymous mutation, which is denoted in blue here, in a minor variant in the right superior lung lobe. Overall, these 2 patients demonstrate that SARS-CoV-2 can infect cells within non-respiratory tissues without requiring changes in the spike genome. In patient 27, which is shown on the left, synonymous mutations were detected both within the respiratory tract and outside of it, specifically in the mediastinal lymph node and the left and right ventricle of the heart. And again, those synonymous mutations are denoted by blue there. While we were unable to sequence many tissues from P33, who was a late case, shown on the right, there were 2 distinct haplotypes detected within the thoracic lymph node of this individual. Finally, we were able to sequence brain samples from 2 patients in our cohort, which are shown here, which revealed non-synonymous mutations between haplotypes within the CNS and the rest of the body, specifically in the dura mater for P36, which is shown here, and the hypothalamus and the thalamus of P38, which are shown here. These results indicate that SARS-CoV-2 within the brain is subject to the development of genetic compartmentalization, and exploring the mechanisms behind this will be important future work. I'll now briefly summarize the histopathologic correlates of SARS-CoV-2 infection in our cohort. Our pathology colleagues in NCI, in addition to running all of our ISH assays, determined the cause of death and characterized the histopathologic findings for all cases. 86% of cases died from COVID, while the remainder died with but not from COVID. Diffuse alveolar damage was detected in all but 18% of cases. A panel shows panel A on the right shows the exudative phase of diffuse alveolar damage with hyaline membranes and mild interstitial inflammation from an early case, and panel B shows the proliferative phase of DID with sparse inflammation in a mild case. Excuse me, not mild, a mid case. Acute pneumonia was present in 61%, and pulmonary hemorrhage was detected in 32%, demonstrated diffusely in panel D of the images on the right, and pulmonary thromboembolism, was detected in 23%, an example of which is in panel C in a medium vessel, pulmonary vessel on the right. That's panel C. We're able to quantify the temporal progression of diffuse alveolar damage from the early exudative phase, which is shown by the orange stacked bars during week 1 to 2, to the intermediate proliferative phase, which are the gray stacked bars, into the late fibrosing phase after 1 month of illness, which are shown by the yellow stacked bars on this figure. In the heart, we observed myocardial infiltrates in only 9% of our cases, despite detection of viral RNA in the heart of 80% of our cases. Patient 3, which is shown in panel E, is an example of a single case that had active lymphocytic myocarditis at the time of death. Bland acute microscopic ischemia and myocardial necrosis was observed in 9% of cases, an example of which is shown in panel F of P38 on the right. We evaluated for the presence of lymphodepletion in lymph nodes and spleen among all cases. Lymph node architecture remained intact without lymphodepletion among 78% of cases. Panels M through O on the right show lymph node of P25, an early case, which is well-defined follicles with marked plasmacytosis and histiocytosis. In contrast, some degree of lymphodepletion was detected in the spleen in all but 5% of cases. Panel K is an example of lymphodepletion in the spleen compared to relative preservation of the white pulp shown in panel J. We found no evidence of direct viral cytopathic effect in the brain, despite detection of viral RNA in 10 of 11 or 91% of our cases. Instead, we observed nonspecific findings, including focal or diffuse hypoxic ischemic injury in 45% of cases. Vascular congestion was observed in 45% of cases, and microscopic hemorrhage was observed in 18% of cases. In summary, we observed that SARS-CoV-2 is widely disseminated across the human body and brain early in infection, that SARS-CoV-2 RNA can persist in multiple anatomic compartments for over 7 months, and we observed a paucity of inflammation or direct viral cytopathology outside of the lungs. With that, I'm going to take a little bit of time to make many important acknowledgments.
The CME code number for those of you taking this credit, it's 37971. 37971.
A really interesting finding that I think that we're focused on, and I think it needs to be focused on, is, why is it that the virus gets to remain in plain sight in all of these extrapulmonary sites in the absence of histopathologic evidence, largely histopathologic evidence of injury and or inflammation? How is it that the virus is eluding the immune response in those extra respiratory sites?
Quick question from me before I get to the next one. The patients where you saw brain infiltration, where, for instance, your Purkinje cell staining, it wasn't clear to me, was that a structural staining to a structural protein of the virus, or was that actually indicating low-level replication in Purkinje cells? Could you?
So the target for our immunohistochemistry was a nucleocapsid protein.
Okay.
So that was nucleocapsid. Based upon, we also, confirmatory assay by ISH. So we approached the brain with a number of additional assays because obviously there are major implications about finding virus in multiple regions of brain and specific cell types. So we're quite cautious about those observations and have taken a number of assays to support or confirm that cell type specificity. As far as replication in the brain, Nihal, we have right now indirect and are pursuing direct evidence of replication competent virus in the brain. The indirect evidence is that the sequencing results that I showed you show evidence of minor variants in the brain, and sometimes not minor variants. Actually, they're nearing equal distribution of variants with non-synonymous mutations that we detected in the brain that we did not detect in the respiratory tissue, suggesting that ongoing viral replication does take place in the brain and that there actually is some evolution of the virus in the brain. We have tried to grow virus from the brain. We're not done. We're going to persist. So far, we have not definitively accomplished that, but it is something that we continue to pursue.
And your answer is related to two questions, so let me ask those. One is from Roland Owens. Do your virus distribution studies suggest any novel receptors? So, for instance, in the brain, what's the distribution of the classical receptors versus what might be novel? And then second question is from Carolyn Ugaru. Thank you for this extremely insightful seminar. Can you please briefly elaborate on the genetic compartmentalization you see in the infected brain tissues? What exactly does that mean? You kind of alluded to that, so maybe you can reiterate and give.
Yeah, that's perfect. Dr. Owens, thank you for your question and also for the second question as well. Specifically, we did not or have not yet queried the association with the known SARS-CoV-2 receptors and co-receptors in tissues and co-localization or presence of a virus by our assays, largely because others have focused on the proteome map of the human body and presence or absence of these receptors in different tissues, and so we didn't pursue that. Obviously, that is something that we could pursue. We cannot directly answer that question, but there are cell types that are not known to have TMPRSS2 or ACE2 receptors. Monocytes are an example, and this has been now recently published in Nature, as I think Nihal realized, she's shaking her head, that there's evidence that those cells are either infected or perhaps take up opsonized virus and are able to accomplish that in the absence of the traditional SARS-CoV-2 receptors. So I think that's work that we can pursue, and that's additional work that likely needs to be accomplished. As far as the compartmentalization of the virus, I do think I showed 6 cases, and I think perhaps the last 2 cases, which are the 2 cases where we had viral RNA that we could query not just from non-CNS sites but also from the brain. In those 2 cases, and I guess I could bring them up, but anyway, in those 2 cases, the important observation, and again, this is an N of 2. I think it's something that needs to be followed up with larger cohorts and cohorts that sort of emulate our study design, that sort of optimized tissue preservation and short postmortem intervals to be able to glean the observations that we've had. But the observation in an N of 2 is that there were viral variants in the brain, populations of viral variants in the brain that were not detected in the respiratory tract, suggesting that there's seeding of a virus in the brain, there's ongoing replication in the brain, and that we obviously know there's an RNA virus that's error-prone, and that as it replicates, there are variant populations that are produced, And through the very sensitive sequencing technique out of the Boritz lab, we're able to detect variants in the brain that were not detected in the respiratory tract, suggesting that there is replication there, and perhaps that there's a particular immune environment there, and this is speculative. Obviously, that's an immune protected site that would allow for viral replication and evolution of the virus.
And related to that, again, there's a lot of questions about the brain, naturally. In the CSF, were you able to find any viral genomes and antibody levels? Were you able to quantify it all in the CSF?
Yeah, good question. The short answer is I don't know. I need to phone a friend from Dr. Stein. So I know as to whether or not we collected CSF. If we collected CSF, it was on a limited number of individuals, and whether or not we detected viral RNA or subgenomic RNA, I cannot recall. I'd have to phone a friend and check our preprint. But what I do know is that we didn't pursue evaluation of antibody responses, or for example, soluble mediator responses, or otherwise in CSF. I'd have to go back to review the inventory to determine what exists that might be accomplished with that sample type.
Sticking with the CNS, have you seen the presence of viral particles in eye tissues? From a Ramakrishna is asking.
Yeah, thank you for that question. So we did query eye tissues. We found presence of viral RNA in eye tissues. That is summarized and categorized in the preprint. What's not in the preprint are confirmatory characterizing cell types in the eyes, and that may or may not be forthcoming in a separate body of work.
This is Thomas Sabotka asked this question, but is there any indication? I mean, this is very speculative, that residual COVID-2 in the brain or other tissues could become reactivated to produce some type of disease condition in the future. I mean, I think it's very early to say, right? I mean, we're at the beginning of this.
So you're correct, Nihal. That is your answer is the correct answer. We can't answer that based upon the study that we did. It wasn't designed as such, but for certain, those are the types of questions that need to be pursued in the future. And obviously, there are concerns or questions about, well, what is the relationship between post-acute sequelae of COVID or PASC and our observations of persistence of viral RNA in many anatomic sites. And again, acknowledging a limitation of our study, we didn't query a PASC population. And so we're cautious about making overstatements about how our findings relate to PASCs. But what it does highlight is that in that patient population, that there should be studies that are designed to specifically evaluate whether or not there is persistence of viral RNA in different anatomic sites, and to what degree that persistence is associated with the clinical manifestations of PASCs.
Thank you. And there's a question from Chase Nelson, I guess relying on what you know about the literature. How likely is it that other respiratory viruses, say common cold coronaviruses, also have similarly widespread tropism and persistence in non-respiratory tissues? Or has it been gone undetected? It's there, but
yeah, that's a great question, and it's one that we have had some really enjoyable conversations and debates around. And the implications are, should we extend this approach to fill in the blank X, Y, and Z respiratory or other viral illnesses that are associated with human severe human illness. What I'll say is this, that part of our approach for CoV-2 was informed by the historic literature of CoV-1, same receptor, MERS, different receptor, a smaller body of literature, but a literature that fairly clearly defines that these coronaviruses, at least the severe human coronaviruses, make their way outside the respiratory tract and infect tissues elsewhere. And so based upon that literature and our sort of understanding, we pursued this quote unquote aggressive approach to better understand CoV-2. If you look at the literature, for example, influenza, I don't think that folks have taken the type of approach that we have to characterize influenza viral distribution across the body and brain. But having said that, people have been studying influenza for a really, really long time and have done a lot of careful studies. And those studies do suggest that the prevailing knowledge and dogma is that those viruses, at least the predominance of them, absent the high path avian influenza viruses that have this special polybasic cleavage site that allow them to get outside of the respiratory tract, that the seasonal influenza viruses that we're infected with don't do this. But of course, you know, if you don't look, you don't know. And so those are ongoing discussions. We're happy to be interested in other people's thoughts, but we are discussing those among ourselves.
And certainly there's none of this anecdotal, at least clinical neurological, evidence that people say they have with, even personally speaking from my, having experienced COVID personally, the brain fog, the vertigo, these things persisted for months after I, tested negative for the virus. So none of those you don't hear from patients with flu or the common cold. It's too many right now that it suggests, really.
It's really tough, Nihal. It's super tough because, there's a literature out there in the non-respiratory complications of flu, but most of them appear to be, not due to direct viral infection in those organs, but due to indirect effects. And you're right, the PAX is a major issue, and now there are, strong data supporting that this is not just a vague constellation of things, that it is related to prior infection. And so beginning to unravel the pathogenesis of that, I think is and obviously the NIH has, I think, devoted over $1 billion in research funding to that particular question.
Right. And can you comment on this recent study in Nature where they had followed these patients with fMRI and seen sort of shrinkage of gray matter? How does that connect?
I love that study. I think that study is fascinating. Strongly suggestive, right? I mean, it's really that study from the UK took advantage of these individuals that were systematically studied by MRI pre-COVID era, and then had the ability to follow the same people post-infection and correlate them with, specific functional MRI regions that were really, really fascinating, and found a strong signal that something's not right, and whether that something is not right, persistence of viral RNA in the brain is contributing to that or not. I think that's something we got to figure out.
Right? I mean, it's so important, so important. There's a question about the sensitivity of your infection assays when you're harvesting virus. Could it be that you're missing out on virus levels because your sensitivity is not there in those ex vivo infection assays?
The short answer is absolutely yes. And that is even though that's a simple answer, it's a hard message to translate to the average listener. We know that cell culture is an insensitive way of detecting replication competent virus, and but it's the standard way, and it's the gold standard way. And so the way to get around this, or to try to quote unquote increase sensitivity to find replication competent virus in samples that didn't grow in cell culture, is you can manipulate your cell culture. You can put in humanized receptors. You can take it into a different system. In other viruses, as you well know, Nihal, people have used immunocompromised mice that are neonatal mice that perhaps are particularly susceptible to viral. So there's other ways but viral culture is an insensitive way of detecting replication competent virus. And so is there likely more than we're observing and claiming? Probably, but we have to work with what we have.